Welcome to Riffyn. In this example, we will show you how to perform variance analysis on an experimental process. Here we'll illustrate this with a very simple cell culture process composed of 3 steps. Culture the cells, wash the cells, and then measure their OD. The objective is to understand how our final OD measurements are influenced by various actions or errors in the first 2 steps, cell culture and wash cells. To answer that, we need to perform an experiment, and we'll begin by looking at a pre-existing experiment on this process that's already been structured for variance analysis. So we open that experiment, variance root causes. And here you will see an instance of the variance process with 8 samples on the cell culture step. And each of those 8 samples is connected to 3 replicate samples in the wash cell step. And in the last case, we have each of those replicate samples are propagated to the OD measurement step, where they are individually measured 3 times using a spectrophotometer. And here you see the 3 individual measurements on each of those washed cell samples. So we have started with 8, split those 8 into 3 replicates, and then measured each of those replicates 3 times for a total of 72 measurements in this experiment. Now we've exported the data using Riffyn's exporting capability, and that export is saved as a CSV, which is you can open in basically any kind of analytical software. In this case, we're using JMP to illustrate. So here we see all of the data from our experiments, and all of the properties, all of the specifications, everything we've measured or set on that experiment is saved as columns, and each column is attached to a step, which is shown on the left in groups. Now we're going to perform a variance analysis on this data, where we want to look at how the optical density measured at the end of the experiment is influenced by different properties or different aspects of the upstream steps. So we make the Y response in this analysis the optical density, and then we select the label of the cell culture step, or the sample label for the cell culture and the sample label for wash cells, and then we perform an analysis. Here you see the structure of the experiment. At the bottom you see the 8 cell culture samples, then the 3 replicates of each of those samples above them, and finally the 3 measurements of each of those replicates. This is a variance analysis, so at the bottom we can see the statistics generated, which illustrates the breakdown of the variance into the 3 steps: cell culture, wash cells, and the leftover variance within. As you can see, much of the variation is introduced at the final step, the measurement of the optical density, which is not where we would really like the variance. So this would suggest that we need to improve our process to reduce the measurement error in that final step in order to get better outcomes. Thanks for watching.